Welcome back everyone. In our last video, we explored the entire pathway of bilirubin synthesis and metabolism. Today, we are moving into a clinically important area that directly relates to bilirubin, that is, jaundice. In this session, we'll try to clearly understand the different types of jaundice and more importantly, how to interpret the key investigations, especially liver function tests, that help us diagnose the underlying cause. Let's get started. Jaundice is the yellow discoloration of the skin, sclera, and mucous membranes caused by elevated bilirubin in the blood. In simple terms, it's not bile or the liver directly making you yellow. It's the accumulation of bilirubin, the yellow pigment produced from the normal breakdown of hemoglobin. Before we dive into, let's first briefly go through the synthesis of bilirubin, which we already discussed in the previous lecture. The senescent RBCs phagocytosed and release heme. The heme, by the action of heme oxygenase, convert into biliverdin, then to unconjugated bilirubin. As unconjugated bilirubin is lipid soluble, so with the help of albumin, it is transported to the liver. And in the liver, the hepatocytes uptake the unconjugated bilirubin and converted into the conjugated bilirubin with the help of enzyme UDP glucuronyl transferase and make it water soluble. Then this conjugated bilirubin is stored in the gallbladder and secreted as bile into the gut. By the action of gut bacteria, this conjugated bilirubin is broken down into the urobilinogen, then to stercobilin, which gives the brown color to the feces and excreted in the stool. The small amount of urobilinogen reabsorbs and excreted into the urine, which imparts yellow color to the urine. In a nutshell, this is bilirubin metabolism. And if this pathway is disrupted at any point, for example, if liver cells are damaged and can't conjugate bilirubin properly, bilirubin begins to accumulate in the blood. This buildup, whether conjugated, unconjugated, or both, is what eventually leads to the yellow discoloration of the skin and eyes. Now let's discuss the type of jaundice, their cause, and the investigations to diagnose it. There are three main types of jaundice, depending upon where the problem lies in relation to the liver. We divide into prehepatic, hepatic, and post-hepatic type. Prehepatic jaundice, as the name suggests, means the problem occurs before the liver. In this type, the issue arises due to excessive breakdown of red blood cells. When a large number of RBCs are destroyed, a high amount of unconjugated bilirubin is produced, much more than the liver can handle. Let's understand this with an example. Suppose the liver can conjugate a maximum of 10 molecules of unconjugated bilirubin at a time. Under normal conditions, there are about 5 molecules of unconjugated bilirubin in circulation. So the liver easily conjugates them. Now, if the body suddenly starts breaking down more RBCs, the level of unconjugated bilirubin rises sharply. The liver tries its best to conjugate as much bilirubin as possible, but it simply cannot keep up with the excess. As a result, the concentration of unconjugated bilirubin in the blood increases significantly. At the same time, the liver does manage to conjugate a bit more bilirubin than usual. So the amount of conjugated bilirubin also rises slightly, though it is still not very high. This conjugated bilirubin is excreted into the bile and stored in the gallbladder. From there, the conjugated bilirubin moves into the intestine, where it is converted into urobilinogen. Some of this urobilinogen is reabsorbed into the bloodstream and excreted in the urine, while most is further converted into stercobilin and eliminated in the feces, giving stool its brown color. Unconjugated bilirubin, on the other hand, is lipid soluble and cannot be directly excreted by the kidneys, which is why it accumulates in the blood during prehepatic jaundice. The common cause of the prehepatic jaundice include the hemolytic anemia, like in sickle cell anemia, thalassemia, because of mismatched blood transfusion, malaria, or other infection causing RBC's destruction. So remember, the prehepatic jaundice means too much production of unconjugated bilirubin due to hemolysis. And investigations to confirm the prehepatic jaundice and hemolysis. The total bilirubin level is increased. The indirect bilirubin is markedly increased. Direct bilirubin is normal or slightly increased, as the problem is not in the liver but due to the excessive breakdown of RBCs. In prehepatic jaundice, the urine color is usually normal or slightly dark, but not the classic cola-colored urine seen in hepatic or post-hepatic jaundice. The slight darkening occurs because of an increased level of urobilinogen in the urine. This happens since there is a mild rise in conjugated bilirubin production. More conjugated bilirubin enters the intestine, where it gets converted into urobilinogen. Some of this urobilinogen is reabsorbed into the bloodstream and then excreted by the kidneys, giving the urine a slightly darker appearance. However, the urine test for bilirubin remains negative because unconjugated bilirubin, being lipid soluble, cannot be excreted in the urine. As for the stool, its color is normal or sometimes darker than usual. This is due to the increased formation of stercobilin in the intestine, which is derived from the excess urobilinogen. In other lab investigations, the lactate dehydrogenase is markedly elevated, as LDH is the enzyme found inside the RBCs when cell rupture. It releases into the bloodstream, so there is elevated level. The level of heptoglobin is low or absent, as heptoglobin is a protein that binds free hemoglobin released from the broken RBCs in the blood to clear it. So in hemolysis, it gets used up, leading to very low level. The reticulocyte count is markedly elevated, as reticulocytes are the immature RBCs. The bone marrow compensate for the loss of RBCs by increased production, leading to high reticulocyte count. The high reticulocyte count is a sign of increased bone marrow activity. The ALT, AST, and ALP level is normal, as no significant liver or bile duct injury occur. In hepatic jaundice, the main problem lies inside the liver. Here, the liver cells are either damaged or not functioning properly, so they cannot efficiently take up, conjugate, or excrete bilirubin. Because of this impaired uptake and conjugation, the level of unconjugated bilirubin in the blood begins to rise. At the same time, remember that hepatocytes also line the small bile canaliculi. When these hepatocytes are damaged or die, they allow conjugated bilirubin to leak back into the bloodstream. As a result, you will see a mixed pattern. Both direct, conjugated, and indirect, unconjugated bilirubin levels may be elevated in the blood. Because the hepatocytes are injured, less conjugated bilirubin reaches the intestine. Consequently, the gut produces less urobilinogen and therefore less stercobilin. The common cause include the viral hepatitis like hepatitis A, B, and C, the alcoholic liver disease, cirrhosis, drug-induced liver injury like paracetamol overdose. So in hepatic jaundice, the problem is in the liver. The investigations in case of hepatic jaundice, the total bilirubin level is elevated, as direct and indirect bilirubin level are elevated, which are the hallmark of hepatic jaundice. The urine color is tea color or Coca-Cola color due to presence of conjugated bilirubin, which is water soluble and excreted in the urine. Urine bilirubin level is positive, as the conjugated bilirubin spill into urine. Urine urobilinogen level is decreased. Stool color. The stool color is pale but not as completely colorless as in post-hepatic because some bile still reaches the intestine but less than normal. The liver enzyme, the AST-ALT, markedly elevated due to liver damage. ALT is more specific to liver in most cases of acute hepatitis. For example, viral. Drug-induced, ALT is typically higher than AST. But in case of alcoholic liver disease, the AST to ALT ratio is often more than 2:1. The alkaline phosphatase is mild to moderately elevated, while a massive elevation is a characteristic of post-hepatic or obstructive jaundice. In hepatic jaundice, the inflammation can compress small bile ducts within the liver, causing moderate rise in ALP level. GGT level are also elevated. Like ALP, it rises with biliary tract issue. An elevated GGT alongside elevated ALP confirmed the source of ALP is hepatobiliary system and not the bone. And also ultrasound shows the fatty liver, cirrhosis, and the parenchymal changes in the liver. So these are the investigation in case of hepatic jaundice. The post-hepatic jaundice, also known as obstructive or cholestatic jaundice, here the problem is after the liver. Here, the liver has already conjugated the bilirubin, but it cannot be excreted into the intestine because of a blockage in the bile duct. This blockage increases the pressure inside the bile duct, causing bile to leak through the tight junctions between the hepatocytes that line the walls of the bile duct. Along with conjugated bilirubin, bile salts, bile acids, and cholesterol also enter the bloodstream. When these substances deposit in the skin, they cause pruritus. There is also hypercholesterolemia and xanthoma formation. Because of the obstruction, no bile reaches the intestine. As a result, there is little or no formation of urobilinogen and stercobilin. Common causes of post-hepatic jaundice include gallstones obstructing the common bile duct, tumors of the pancreas or bile duct leading to obstruction, biliary strictures, post-surgical obstructions, primary sclerosing cholangitis, and biliary atresia. On investigations in post-hepatic jaundice, the total bilirubin is elevated. The indirect bilirubin is usually normal, while the direct bilirubin is markedly elevated because conjugated bilirubin backs up into the bloodstream. The urine becomes cola-colored or tea-colored due to the presence of conjugated bilirubin in the urine. Urine bilirubin is present as conjugated bilirubin spills into the urine. Urine urobilinogen is decreased or absent because no conjugated bilirubin reaches the gut. The stool becomes pale or clay-colored because no bile enters the intestine, so no stercobilin is formed. The markers of biliary obstruction, such as ALP, are markedly elevated. ALP is an enzyme present in the cells lining the bile ducts, and obstruction increases the synthesis and release of this enzyme into the blood. So ALP levels rise significantly. GGT is also markedly elevated since GGT is highly concentrated in the biliary system, so its levels also increase. AST and ALT are normal or only mildly elevated because the primary problem is obstruction, not hepatocellular damage. Imaging studies like abdominal ultrasound typically show the obstruction, such as stones, strictures, or tumors blocking the bile duct. So in post-hepatic jaundice, the bile flow is obstructed after the liver.